We often picture a eukaryotic cell as a simple, water-filled balloon holding a few floating parts. But if you pass directly through the plasma membrane, you find a densely packed, highly organized metropolis. Pushing through the cellular fluid and approaching the nucleus, you hit a massive, labyrinth-like structure that completely surrounds the genetic center. Discovered independently in 1945 by researchers Keith Porter and Ernest Thompson, this is the endoplasmic reticulum, or ER. Its scale is immense. The interior space of this organelle, known as the lumen, regularly consumes more than 10% of the cell's total volume. Architecturally, it functions as a continuous, branching pipeline that connects directly to the double-layered nuclear envelope and extends outward deep into the cytoplasm. At just 5 to 6 nanometers thick, this membrane network provides the critical infrastructure the cell uses to manufacture, process, and transport the materials it needs to survive. The ER is divided into 3 interconnected zones: the nuclear envelope ER, the rough ER, and the smooth ER. The membrane takes 3 forms: flat cisternae, round vesicles, and branching tubules. The rough ER is built from flat, interconnected cisternae stacked tightly against the nuclear envelope. It's classified as rough because the surface is studded with RNA-rich ribosomes. These attached ribosomes use the rough ER as a base of operations, synthesizing proteins and translocating them directly through the membrane wall and into the internal lumen. Once inside, the rough ER acts as a strict quality control checkpoint. If a protein is folded improperly, it is rejected and exported out to cytosolic proteasomes, specialized cellular recycling complexes, for immediate degradation. The internal spaces of this network are in constant dynamic motion, with newly formed proteins rapidly navigating the tight corridors. By combining the architecture of flat cisternae with the machinery of ribosomes, the rough ER functions as the definitive protein factory of the cell. Further out from the nucleus, the membrane smoothly transitions into the smooth endoplasmic reticulum. It forms a continuous system with the rough ER, but it completely lacks attached ribosomes. This table compares the structure of the 2 networks. Notice how the smooth ER lacks surface ribosomes and pores, relying heavily on a network of tubules and vesicles instead of flat cisternae. Free from protein synthesis duties, the smooth ER handles intense chemical processing. It drives lipid and steroid biosynthesis, producing critical molecules like cholesterol, estrogen, and testosterone. In liver cells, it acts as a primary drug detoxification center. Here, the smooth ER uses cytochrome P450 enzymes to add hydroxyl groups to incoming toxins, making hydrophobic or water-repelling drugs soluble and easier to excrete. In muscle cells, it takes on yet another role. Known as the sarcoplasmic reticulum, its lumen stores exceptionally high concentrations of calcium, releasing it on command to trigger muscle contractions. Normally, cellular membranes achieve high curvature because their lipids are distributed asymmetrically, but the smooth ER doesn't use that mechanism to hold its highly curved tubular shape. This schematic diagram details how proteins manipulate the structure directly. Reticulons act as shaping proteins to actively bend the membranes into tight curves, while atlastins drive the fusion of these tubules to build a continuous, 3-way polygonal grid. The smooth ER abandons the protein-building machinery of the rough ER in favor of extreme structural curvature. That physical adaptation allows it to manage complex lipid chemistry, neutralize toxins, and regulate cellular signaling. The ratio of rough to smooth ER inside any given cell isn't arbitrary. That specific balance dictates the cell's physiological role in the human body. This illustration and corresponding electron micrograph of a mucus-secreting goblet cell show a distinct polarization of organelles. Notice the basal end at the bottom. It is densely packed with a massive array of rough ER, specifically configured to synthesize massive quantities of mucoproteins. Compare that to the lighting cells of the testes, or the cortisol-producing cells of the adrenal glands. Because their primary job is to pump out steroid hormones, their internal space is dominated by a vast smooth ER network. The ER is a constantly shifting, dynamic factory that reacts and adapts to the biological needs of the organism in real time. It is the ultimate cellular adapter. Whether a cell builds tissue, stores memory, or neutralizes poison, that capability is directly defined by the physical morphology of its endoplasmic reticulum.